Thank you for the invitation. I will present our work on the modeling of gene regulatory network. I entitled my talk The Biologic of Regulatory Networks, and I think that there is some work to do in understanding how to evolve these gene regulatory networks to ensure some function. So this will be probably a topic to be discussed later on. So the main motivation is that we know that there are regulatory networks, interaction networks, underlying the functions of biological processes. So for example, here we have the well-known cone map driving the cell cycle control. So my interest is not only to study these networks, but the dynamics driven by these networks. So the idea that from a biological process, here the segmentation of the Drosophila embryo, for example, we can delineate a model. First, the network of the interacting genes, for example, that are known to play a role in this biological process. And then to study from the model the dynamics and more precisely the properties of these dynamics. The first question is about the asymptotical behavior. So we know that these networks, for example, will drive the system to specific attractors, in this case of the Drosophila embryo, in specific pattern of expression. Then we can ask also the properties along the trajectories going to a given attractor. From an initial condition, what are the conditions to drive the system towards a given attractor? What are the role of the input components? We know that there is an influence from, for example, the environmental signals or neighboring cells. What are the influence of these input signals? We can study also the influence of perturbation. If we have a gene knockout, for example, what is the impact on the dynamical properties? Finally, we know and we can study, proposed by René Thomas, where we have Boolean or extended multi-valued variables. Sometimes you need more than on-off abstraction. You need additional threshold, and also by the fact that we have an associated delay between the time where the gene, for example, received the order to be expressed and the production of the protein. This will come more clear in the next slide. So what we call a logical model of a regulatory network is defined by an interacting or a network where we have two kinds of arrows, the positive one which will convey an activation effect, and the red arrows which are inhibitory effects. Each node represents a regulatory component, a gene, a protein, or even a phenomenological node, and it is associated to a variable which can be in general a Boolean variable. And then we define logical rules that convey the regulatory effects of the regulators towards a node. For example, A has a unique regulator X, and we can say that it is on if X is off and is off otherwise. And the same for C, for example, where it's a bit more complicated because we have the combination of interactions here from B and D, and then we have to decide how this regulation will combine to drive the dynamics of the variable associated to C. So a question, for example, if I give an initial state of this network where only X is expressed or present and all the other nodes are absent, what is the future of the system? So we can say from the logical rules that B should increase its level and X should decrease its level. So it's quite intuitive. But then we can represent these dynamics by another graph, which we call a state transition graph, where we have the nodes representing the states of the system. And from this initial state, 0, 0, 1, 0, 0, we have two possible events. And because we don't have access to any information about delays, we will generate both transition, both possible events. Either X is turned off or B is turned on. So we have all the potential trajectories of the system driven by the model. So on the left, you have the asynchronous update, where we consider that in the lack of information of delays, we will generate all potential trajectories. And this is the synchronous update, where we consider that all changes in the values are made synchronously at the same time. And you can observe that the behavior and the attractors of the system are quite different. With the asynchronous update, we have a cyclical attractor or a single stable state. So if X is faster, then the system will end up in a stable state. Otherwise, if B is turned on before, then the system will be trapped in a cyclical attractor. In a synchronous update, we have another cyclical attractor, which is quite different from the one on the left. So this is just to say that the decision or the choice of how you update your system is quite important. So the problem with these simple models, even if it's a high abstraction of the reality, it's still complicated when we want to deal with larger networks. So here is the whole potential dynamics on the whole state space of this simple model here. And you can see that it's already complicated, but what we can say that we face a combinatorial explosion problem, which is quite classical, because the number of states explodes and grows exponentially with the number of components of your network. So my group is interested not only in defining models in collaboration with biologists, but also to define methods to tackle the complexity of these models and to be able to delineate their properties. So we have two kinds of methods. The first one is to try to infer properties from the model without having to generate the whole dynamics. This can be the stable state identification or the study of regulatory circuits, and I will say a few words about this. Or from the dynamics. This means that you generate the dynamics and you try to assess the properties of the dynamics. So here we develop a number of techniques or methods to try to define or to assess the properties in a more efficient manner. So I said that we rely on the regulatory circuits, and I also evoked the fact that in these networks we have significant motifs which drive the dynamics, and we believe that regulatory circuits are really important. In fact, it is proved that the positive circuit is required for multi-stability, different stable states or different cyclical attractors. Positive circuits are defined by circuits where you have an even number of inhibition. So along the circuit here, A has a positive effect on itself. And you can see here that for this simple positive circuit, we have indeed two stable states, where negative circuits are required to observe sustained oscillation, and negative circuits are defined by an odd number of inhibition here. And the dynamics of this simple circuit, negative circuit, indeed leads to a cyclical attractor. But these are necessary conditions, and we know that our circuits are embedded in larger networks, so we would require a sufficient condition or how these circuits indeed embedded in networks can drive the dynamics. For example, here we have a negative circuit. On the left, it is what we call functional. We have the cyclical attractor, where here, because we have an external regulator, which will prevent the whole effect of B, we have some zone in the state space where the circuit is functional. We have a sustained oscillation. But when C is present, the circuit is no more functional. We have a stable state here. So we can define, and indeed we develop an algorithm to define the zone or the region of the state space where the circuit is functional and drive the dynamics in the expected way, either creating a separatrix with multistability, if it's a positive circuit, or generating some sustained oscillations. So another example, a bit more complex, where we have here 3 nodes, and C here is multivalued, saying it can take value 0, 1, or 2. And here I have a positive loop on A, a self-activation, and I put here the functionality context, meaning that this positive loop creates a separatrix in the state space here, whenever B is 0, so in this region, or when B is 1 and C is 0, which is this part of the state space. So here we can see the role of this auto-activation. For this negative circuit, it is functional provided the absence of A, and we can see here the sustained oscillation driven by this negative circuit. Now, if we change the rule, which means changing only the rule driving the dynamics of B without touching the wiring of the system. So we have still the same set of positive and negative circuit. Now the only circuit which is functional is this negative circuit, and it is functional in the whole state space. And we can see that the unique attractor in this case is a cyclical attractor. So I just wanted to present this qualitative framework to model complex gene regulatory networks. And this formalism can convey any kind of regulation. So here we have only the semantic of influence between nodes. So what is a regulation can be a transcriptional regulation, but it can be also phosphorylation of a protein. And we know, for example, in the cell cycle control, we have a lot of control from based on protein phosphorylation. And I wanted to quote this sentence from Adrien Fauré and Denis Cheffry, who made a review on comparing the models, qualitative models of cell cycle control. And they say that beyond homology relationships between the regulators, which could be understood as the wiring of the network controlling the cell cycle, mathematical models of the cell cycle highlight the conservation of the regulatory circuits. And I think this point is really important also to consider the motifs and the dynamics related to these motifs. So just to conclude, this qualitative modeling framework, which can be useful to convey the role of different structures in gene regulatory networks to drive the dynamics, the importance of the update of these logical models. A lot of work has been done on synchronous updating, but I showed that this can have an impact on the properties of the dynamics, and the crucial role of regulatory networks. So just some acknowledgements and of all my collaborators and the group at the IGC. I thank you.